Adrian and Malachi, please let me know if you need assistance taking the exam. It is on SchoolNet. I will send the test code to the chat so that you have access to it as we move on.
I'm doing it now.
Okay. So for everybody else, the title of today's lesson is Characteristics of Life. Today's date is December 4th, 2020, and it's the first day of our last unit. We will have 5 days, so I guess next Friday is going to be solely dedicated to review, and you all will have time for makeup work. The objective of today's lesson is to describe the characteristics of living organisms and the essential question, how are the STERNGRR processes dependent upon one another? So this is a concept that I promised way back in August we would be coming back to, because STERNGRR are the 8 essential life processes. And if you can really understand these 8 essential life processes, you're in a great place to do well on the test, because they do cover almost everything we've talked about this semester. All right, so going back to Unit 2, all living things are made up of what? What's the basic unit of life?
DNA.
Okay.
Oh, no, cells.
Good. Thank you. All living things are made up of cells. All living things have DNA and RNA to hold what? What does DNA and RNA hold? What does it store?
Genetic code.
Thank you, Alicia. Genetic information, or the genetic code. All living things have to make proteins through a process of protein synthesis, which we'll talk more about in the next slide. But the process of protein synthesis involves transcription of DNA to RNA, and then translation of RNA into amino acids. All living things finally have to complete essential life processes in their own unique way, and that is STERNGRR. All right, so here's a flashback of protein synthesis. This was the 5th unit, the unit right before our midterm exam. DNA is going to be transcribed into RNA, or mRNA, messenger RNA. That's the process of transcription. So keep in mind that these processes, they're easier to remember if you can see that they occur in alphabetical order. Transcription is first, and translation follows it. DNA is transcribed to mRNA. mRNA is translated into amino acids. Those amino acids will eventually form into a protein, and proteins are what give us our traits. They are what give living things their traits. So the process has to start with DNA. If you change the DNA, then you're going to change the traits, because you're going to change the proteins that can be made. All right, so quick practice question. Which most directly affects the sequence of amino acids in a protein? A, the DNA in the nucleus of the cell. B, the amino acids found in the cytoplasm. C, the type of cell in which the protein is made. Or D, the region of the cell in which the protein is made. Which one most directly affects the sequence of amino acids? Go ahead and send your answer to the chat. Okay, I've got an answer from Alicia. Anybody else want to share their thoughts? Malachi, you should be taking your test, but I appreciate you. Joseph, thank you. Castro, thank you. Dante, good. All right, so everybody's jumping on the A wagon. The A wagon is the correct wagon.
I chose another answer, but everybody kept saying A, so I didn't want to be wrong. So
Okay, groupthink took over here, but A is the correct answer. The reason is that, as I was just discussing, this process of protein synthesis starts with DNA. If you change the DNA, you ultimately are going to change the proteins that are made. All right, you're changing the sequence of the amino acid. The DNA is the recipe book. If you change the order of the steps in the recipe, or you change the ingredients in the recipe, you obviously end up with a different meal. We've got to think about DNA and proteins in the same way. Now, I've always thought about it in this way. Let me send it to the chat. DNA is the recipe. Amino acids are the ingredients. Proteins are the meal. So DNA tells you you need to put this ingredient inside this ingredient. You need to take this ingredient and heat it up. You need to connect these two ingredients. DNA is telling you, it's the recipe that's telling the amino acids what to do and how to assemble themselves. If you assemble the amino acids or the rest, if you assemble the ingredients in the right way, if you follow the instructions of the recipe, then eventually you're going to end up with a meal. You're going to end up with a correct protein. So that's the analogy that I like to follow. But if you think about the first step is having the recipe in the first place and having the correct recipe. If you change the recipe, even in small ways, you can have profound impacts on the outcome of the meal. If you say add a cup of water and you meant to say add a tablespoon of water, that's going to totally throw off the end results of the meal, even though it's a small change. Okay, so the DNA is important.
I did that with brownies once by accident.
And how did they turn out?
They were all liquidy, not liquidy, but they weren't, they weren't how it's supposed to look.
Yeah. Yep.
They
See,
were different.
we've got a real life example. Okay. Second practice question: how many amino acids are affected by the deletion of one nitrogen base? Is it A, all amino acids are affected? B, no amino acids are affected? C, all amino acids before the point of the change are affected? Or D, amino acids from the point of change and beyond are affected? Go ahead and send your answer to the chat again, please. Okay, we've got an answer from Dante. We've got an answer from Alicia. A half answer from Alicia, kind of. She's given us two answers. We've got an answer from Ayana and Olivia. Okay, good. So the D's are starting to roll in, and the D's are correct. So I'm going to demonstrate this on the next slide. But when you change when you change the amino acid sequence, meaning that you insert something or you delete something, it throws off everything that comes after that point of insertion or that point of deletion, everything that comes after that. Let's see how this works, because it's called a frameshift mutation, and it's a pretty important concept. So let's say that I have my DNA on the top, and my DNA sequence is TACGGCCGT. When I transcribe that DNA into mRNA, I end up with AUG CCG, GCA. Now, hopefully you all remember this. This was something that we did practice quite a bit several months ago. But remember, when we transcribe DNA, T is transcribed to an A gets transcribed to a U, because there are no T's in RNA. No T's in RNA. C and G are pairs. So C is transcribed to G and G is transcribed to C. So this is what I end up with in my mRNA. Then I have to use a codon chart or an amino acid chart to translate my mRNA into amino acids. So AUG is a codon. A codon is a group of 3 mRNA nucleotides. So AUG is a codon, CCG is a codon, GCA is a codon. They're groups of 3. I take this AUG and I use my codon chart to figure out what the corresponding amino acid is. So my first letter is A, so I know I'm looking in this section here. My second letter is U, so I know I'm looking in this section right here. And my third letter is G, so that tells me that I've got the amino acid methionine. And then I keep going, CCG. CCG is proline. And then I get GCA. GCA is alanine. All right, now, but watch what happens. Let's delete the 4th base. So this is what just happened in our previous practice question. We deleted one of the nitrogen bases. So let's say we delete the 4th one in this case, the guanine nucleotide, that first G. I delete it. Now look what happens. My first amino acid is not affected. We still have the same codon for the first amino acid. It's not affected because it happened before the deletion. It took place before the deletion. So I still end up with methionine first. But then now that I've deleted this G, then I now have to shift everything over. My reading frame is now shifted. So instead of having CCG as my codon, I now have GGG. Oh gosh, I can't talk. CGG. Do we see how it was shifted over? Instead of CCG, now I have CGG because this got deleted. And that throws off my amino acid. I did have proline, now I have alanine. Then my next codon was supposed to be GCA, but look what it will be now. It's CA, whatever comes next. So everything that takes place after this deletion is going to be thrown off. Instead of methionine, proline, alanine, now I have methionine, alanine, something else. All right, so frameshift mutations are extremely harmful. Somebody else just showed up. Who was it? Angel. Is Stephanie here? No. All right. So a recap of STERNGRR. STERNGRR is an acronym that helps us to remember the 8 essential life processes. And this, if I were you, would be kind of the foundation of my study guide as I prepare for the EOC. I would break down each of these terms and see how many other concepts I can relate to them. For example, synthesis is the use of small things to make bigger things. So when I hear small things, maybe I should think about monomers. And I know that monomers, when we combine them, become polymers. For example, the monomer of a carbohydrate is glucose. If I combine glucose together, I end up with a much larger sugar. When I'm thinking about synthesis, maybe I'm also thinking about enzymes. Enzymes help to build up, they take two substrates and they combine them into one to end up with a singular product. Or I could think about the example that we have on the screen, photosynthesis. When I'm taking carbon dioxide and water and sunlight to create glucose. All right, so if I were you, I would really use STERNGRR as the foundation of my study guide. In fact, I might take a screenshot of this screen, or I might take a picture of this screen, and I might have each one of these letters their own page in my notebook, and see how much of this unit, how much of this course I can relate to it. Reproduction, I can relate the cell cycle to that. I can relate fertilization to that. Growth and development, I can also relate the cell cycle to that. All right, so there are all these cross-cutting concepts that we've gone over the last 4 months or so, 3.5 months, and they all come back to STERNGRR. That's why we introduce it in the very beginning, and that's why we come back to it at the very end of the course. All right, let's see.
Who teaches chemistry?
This year, Miss Fields teaches it, and Dr. Rosenlind, she's new to Westmec. This year, those are the two chemistry teachers. "29 questions, let's do it." It's gonna take a little while, but we have nothing else to do except for the asynchronous assignment. All right. www.kahoot.it 7599072. All right, thank you, Jalen. Thank you, Olivia. Thank you, Alicia. Thank you, Ayana. Thank you, Omari. Thank you, Malachi. Thank you, Castro. But there are still several other people who have not joined the game. So I'm going to wait 30 more seconds. 30 more seconds. Let's go ahead and start. Which of the following is not a function of proteins? Proteins do a little bit of everything for our body, except for one of these things that you're seeing on the screen. Yeah. Ooh, this is a tough one. So yes, proteins help to fight disease. They help to control the rate of reactions, and they are used to form bones and muscles. They are the soldiers of our four biological compounds, but they don't help us to store and transmit heredity. That is a job of DNA. Nucleic acids take care of that for us, not proteins. Good job, Alicia and Dante. Enzymes affect the rate of reactions in living cells by changing the speed of the reaction. Good. Which of the following organic compounds is the main energy for living things? All right, so lipids I could understand. Lipids do provide some energy to living things, but carbs, carbohydrates, sugars, those are the main source of energy for living things. All right, nucleic acids, good. I'm glad no one chose that. Proteins, for some reason, there is this idea that proteins provide energy to our bodies. They are not an energy source, but they are necessary for health. So I think part of the reason we struggle with that question is because just commonly we think that proteins are an energy source, and that's not necessarily scientifically correct. The most abundant element in most living things. Please get this one correct. Come on now. Oh, wow. All right, so oxygen is present in all living things. It's a part of all four biological macromolecules, so I could see that. But carbon is so much more abundant. Carbon is what we consider the organic element because it's present in all living things. It's essential to life, and it just shows up far more often than oxygen does in greater quantities. Good job, Alicia and Castro. A monosaccharide is a blank. A monosaccharide is a carbohydrate. Yes, a monosaccharide is a sugar. It means one sugar. Mono is one, saccharide is sugar. Let's go. A single unit that joins together to form a polymer is called, I was just talking about this. Good, a monomer. We join together a monomer and it creates a polymer. Substrates fit into the blank blank of an enzyme. 2 words. Oh, all the answer choices are 2 words. Good, the active site. Substrates fit into the active site of an enzyme, and that's where things get active. Good job. What is the monomer of a carbohydrate called? Good. The monomer that makes up a protein is called a or an. Amino acids, good. Amino acids make up proteins. Let's see what's next. A glycerol and 3 fatty acids make up a. Good, lipids. That was a little more tough, it looks like, than some of the previous questions. But lipids are the polymer, and then the monomer would be glycerol and 3 fatty acids. What is the optimal temperature for this enzyme? Excellent. So the media, show media. Let's see. We can see that the highest point in the graph is always where the optimal temperature, in this case, and in some cases pH, will be reported. So the graph, the curve is at its highest right here, and that corresponds with 40 degrees Celsius. DNA and RNA, blank. What are they? They're both nucleic acids. Why did so many people say proteins? The NA in DNA and RNA stands for nucleic acid. Curious. Starches are an example of what? Starches are indeed carbohydrates. Excellent. Fats and waxes are they're both lipids. Good. Oils excellent. Lipids, they're also lipids. Another one, sugars are an example of yep, sugars are carbs, carbohydrates. Said that already. Good. Let's keep going. Biomolecules contain atoms of this was an obvious. Somebody said birds. That must have been a mistake or a joke. Biomolecules exist in yeah, we find biomolecules in all living things. That's why they're called biomolecules. Enzymes blank the activation energy of a chemical reaction. What do they do to the activation energy? All right, so some people said increase, but the correct answer is lower. That's how they speed up reactions. They lower the activation energy. No. Blank are catalysts. Enzymes are catalysts. The word catalyst means that it speeds something up. Okay, 9 questions left. Enzymes are very specific and only with certain blank. I believe that first with should be work or fit. Enzymes are very specific and only fit with certain blank. They only fit with certain substrates. Structure determines function. Their shape has to match. Their shapes have to match. Enzymes blank chemical reactions. Okay, this is getting a little bit redundant.
You make these?
Who made this? Potentially one of the other biology teachers. They speed up chemical reactions. A substance that increases the rate of chemical reactions without any permanent change. All right, this is. Okay. Good. Catalysts, aka enzymes. Is the substrate blue or white in this image? Good. The substrate is blue. The enzyme is white. The enzyme doesn't change. I guess this image is not that was not really the most fair question, but typically the substrate looks like it's fitting into the enzyme. Okay, this is silly. All right, not sure why 2 people got that wrong, but 4 more questions. What is this a picture of?
can't see.
You can probably see enough. I'll give you a hint. It has a backbone and some tails coming out of the backbone. Lipids. Yep. What is this a picture of? So I guess those will be the last 4 questions. Carbs. Yep, that was the 6 carbon rings. That was a carbohydrate. All right, which of these is your favorite holiday? I assume that they are all correct answers, so just go ahead and say something. If your favorite holiday is not here, then I apologize. Last question. At what pH will lipase denature? At what pH? Four and 12. You can see lipase is the solid black line. At a pH of 4 and below, it is totally denatured. There's no enzyme activity. And then at a pH of approximately 12 and above, there's also no enzyme activity. Castro, 17 out of 29, pretty good. Ayana, 23 out of 29, really good. And Alicia, perfect. Yet again, 29 out of 29. Good job to all of you. We've got 15 minutes left. Oops, I clicked on it again. And in that time, where's my mouse? I'm going to skip this for now. In that time, I want you all to go ahead and knock out this EOC prep assignment, number 1. It should be quite easy. Well, not easy, but you should have plenty of time to do it. 15 minutes should be plenty of time. So go ahead and knock it out. I'm going to use this time to grade some Unit 9 stuff. If you know that you still have Unit 9 stuff, please do still turn it in. And I will be here on mute.
My fault. I accidentally left.
Mr. Rudd,
Yes, ma'am.
how are we supposed to answer number 5? Because there's no text box.
And which one are you looking at?
The Unit 1 review.
There's no drop down. It should be a drop down. Hold on. It's weird. All right, hold on. Okay, I think I've fixed it. You may need to refresh. Hopefully you don't lose all of your answers. I apologize about that.
It still isn't there for me.
And did you refresh the page?
I refreshed it and then I left the page and then I went back in.
Okay, I think it was because I didn't click save. All right, let's try it now. Hopefully that was it. Oh, and it is 11:35. So I apologize for that mistake. I think it should be fixed now. I think the problem was that I had